Hello, Mr. Allman here, and this is your Allmancast Introduction to the Photosynthesis Lab. Your goal here is to be able to conduct an experiment in which you test the effect of a limiting factor on the rate of photosynthesis. The limiting factors include the temperature, the amount of carbon dioxide available for the leaf, and finally the intensity of light. So consider between those options when you're deciding on your independent variable. There are other possible factors, but those are the ones that you'll see more on, so take a look in your textbook if you want some more background on that. The purpose of this video is to give you just a brief introduction into some of the equipment and some of the options that you should be selecting from when designing your lab. The goal here is not to be comprehensive. I'm going to give you just a little glimpse at what some of those different experiments could be like. When you decide on a particular topic and a way of being able to measure photosynthesis, you're going to want to look at some of the resources that I provided to see some of the details about actually coming up with a valid procedure. One of the items that works regularly and I use every year is spinach. Spinach leaves are readily available and they, when they're fresh, will photosynthesize. The simplest setups will involve taking a large number of leaves, spreading those out across a chamber, exposing them to a very bright source of light, and then looking for the presence of gas exchange. So in a sealed container, if photosynthesis is happening, since the leaf is going to be using carbon dioxide as a reagent in photosynthesis, a carbon dioxide sensor can be used to monitor what the concentration of CO2 gases in that container are going to be over time. And using the LabQuest, I can get the rate of which CO2 is going to be consumed as a measure of photosynthesis. Alternatively, but with the same setup, a Vernier oxygen sensor will measure the percent oxygen gas in a closed container. And oxygen is produced as a byproduct of photosynthesis. So under intense light, if the leaf does photosynthesize, I should see an increase in the amount of carbon dioxide gas, in the amount of oxygen gas. Now, working in that setup, there is a considerable amount of time that's required in order to be able to get that noticeable change in oxygen and carbon dioxide. And the conditions really need to be correct in order to get the intended results. An alternative, but still using spinach, is to be able to take those leaves and to be able to make small little leaf cutouts using a hole puncher. These small pieces of leaf can be submerged into a container that contains baking soda as a stand-in for the amount for a source of carbon to use for photosynthesis. And when you follow a particular procedure using a syringe and negative pressure, you can get those leaf discs to sink down initially to the bottom of your beaker. And then again, expose those leaves to intense light, and you're going to get them to float. And so you can measure the rate of how quickly it is that they can float depending on the condition that you're trying to change. Here with a liquid environment as opposed to a gas environment, that's going to make it easier to do things like change temperature, because changing the temperature of water, which will heat and cool slowly, is going to be a little bit easier for you. Aside from spinach, another photosynthetic material that I have available for you are these. These are algae beads. So algae is an aquatic organism that photosynthesizes, and algae grow on the surface of water in all types of ecosystems, both marine and freshwater. These beads utilize sodium alginate to be able to make a gel, and the algae is trapped inside those beads. This provides a nice, easy way to measure out a specific amount of algae to be able to use in an experiment. The way to use those algae beads is to place them in a cuvette and fill that cuvette with a pH indicator, a very specialized bicarbonate indicator that will change color based on small changes in pH. Because as the algae photosynthesize, they're going to consume carbon from the water, and that's going to cause the pH to be able to actually increase, because more carbon in the water actually forms carbonic acid. Once you note those color change, you can look at the rate of color change and compare to a standard scale for this particular indicator, and that can tell you what the pH is based on the color that it becomes. You are welcome to choose other methods as well, bringing in your own aquatic plant or bringing in your own samples to be able to use as part of this experiment. But those are some of the materials that I've seen year after year students use successfully as part of their lab work. A few other things to consider. If you're testing light, you want to think about how it is that you're going to actually measure how much light there is. And so being able to use a light sensor is one way to be able to measure light intensity, because the light sensor, when pointing it in the direction of the light source, I can actually tell how bright the light is that is reaching my sample. Other things that change about light include not only intensity, but also wavelength. That's a second property that we will learn about in this unit that impacts photosynthesis. The color of light. We interpret different wavelengths with particular colors, and plants also utilize different wavelengths of light differently. And so understanding what the source of wavelengths are, or if you're changing that, the type of wavelengths, those can certainly impact the rate of photosynthesis. A few other things to consider. We're working with living things here, and that's going to increase the number of confounding variables that you have. Obviously, you're going to try to control as much as possible in designing this experiment, but there are some maybe new conditions that you might not be used to thinking about that are certainly going to impact. When we deal with living things, knowing like that every living thing is unique. The DNA in each of these spinach leaves they come from different plants. We have to assume they were picked at different times. They have different sizes. They have different amounts of water in them. And so we're going to expect that with all of these differences, that one leaf is not necessarily going to act exactly the same as another leaf. And two things that will certainly impact them quite a bit in this experiment when using the spinach is going to be how old they are, so how long it's been since they've been picked and how long they've been stored in the refrigerator. And then secondly, how much water they have. Is the leaf, is it droopy and wilted because it's dried out? And that's not going to be very good for photosynthesis. All of these living things, they undergo not only photosynthesis, but also respiration. And so that's another thing that sometimes can be confusing, is that when the leaf is in the dark, or even when the algae beads are in the dark, they don't continue to carry out photosynthesis, but they actually do the opposite. They will break down stored nutrients and produce carbon dioxide gas as opposed to consuming it. And so we might see changes in CO2, the opposite of what it is that we might have expected. All right, so please choose from among some of those options. And again, for each of these different types of methods that I have mentioned here, I've got numerous other follow-up resources that you can take a look at as you decide how it is that you're going to design your lab. Thank you.